on pediatric cell and gene therapy. We are delighted you're here, especially if you're joining us for the first time. My name is Angie Price. My name is Angie Price. I'm the Director of Education and Marketing Communications here at IACT, and I will be your moderator for today's webinar. I look forward to spending the next 60 minutes with you exploring the current landscape of cell and gene therapy and the challenges and considerations when conducting these type of trials. Before we get started, I'd like to cover a few housekeeping items. As you are aware, we are recording the webinar today. A copy of the recording and slides will be provided. You may also access them on our site network portal. The link will be posted in the portal throughout the webinar and shared on the chat. Speaking of the chat, we encourage you to post questions and comments. We will try to get to as many questions and comments as we can as time allows. For those of you that don't know about IACT, we are a not-for-profit organization. We work with patients, sites, and pharmaceutical companies to expedite pediatric drug development. We support a network of over 70 trial-ready sites by providing clinical trial opportunities, mentoring, quality improvement initiatives, regulatory support, and educational webinars like this one. We also work with pharmaceutical companies on pediatric clinical design and feasibility, site identification projects, and initiatives to advance pediatric research. Our vision is to reduce the time it takes for pediatric drug development from 9 to 5 years because children can't wait. And now it is my pleasure to present our speakers. We will begin with Dr. Russell Cruz from Children's National Hospital. He will begin by telling us a little bit about himself. Welcome, Dr. Cruz.
Hi there, everyone, and thank you, Angie. Good morning. So my name is Russell, and I'm a translational immunologist with over a decade's experience in cell and gene therapies. So my work right now is focused on developing immune cell-based therapies for opportunistic infections like HIV and refractory brain and solid tumors, using novel engineering approaches, including gene modifications, nanoparticles, and small molecule inhibitors. I am an associate professor of pediatrics and molecular and microbiology, immunology, and tropical medicine at Children's National and George Washington University in Washington, D.C. And education and public outreach is among my key interests. So looking forward to learning with everyone here.
Thank you, Dr. Cruz. We're very fortunate to have you here today. And now we will meet Dan Eisenmann from Advarra.
Hi, I'm Dan Eisenmann, Executive Director of Biosafety Services at Advarra. I've spent my career in the field of biosafety and head Advarra's Institutional Biosafety Committee Review Service that focuses on assessing the risks associated with engineered genetic materials, which includes gene-modified cellular therapies, genetic vaccines, gene therapies, and gene editing, and PhD in molecular biology and immunology. And I frequently speak at conferences and at webinars, as well as publish peer-reviewed articles on this field, and I'm very much looking forward to participating in this webinar.
Great. Thank you, Dan. So happy that you could join us today. Finally, we have Sarah McCague from Children's Hospital of Philadelphia. Welcome, Sarah.
Hi, thanks, Angie. My name is Sarah McCague. I am the Administrative Manager of the In Vivo Gene Therapy Group at Children's Hospital of Philadelphia. I started at CHOP almost 13 years ago as a research coordinator in gene therapy, and I never left. And now as the manager of CIGT, we're working to streamline clinical gene therapy at CHOP. So helping investigators and also helping to deploy commercial approved gene therapy products. Excited to be here.
Thank you, Sarah. We're so happy to have Sarah to be able to provide the site's perspective a lot on the conduct of cell and gene therapy. You know, I want to express my gratitude to the speakers for joining us today and sharing their expertise in this growing field. I know that their time is so valuable, and your commitment to pediatric education is truly commendable. So thank you for being here. I would now like to turn it back to Sarah. She will be sharing firsthand the profound impact gene therapy can have on a child's life.
Thanks, Angie. So I think, first of all, I just want to address why gene therapy? Why are we here today? Why are we talking about this? Why is this important? And so I'm going to start with a story that's close to my heart, which is the story of Luxturna. When I started at CHOP, as I said, I was a research coordinator, and I worked on a clinical trial for a type of inherited blindness. The drug was later named Luxturna, and it became the first approved product, first in vivo gene therapy product in the US. So on this slide, there's a picture of me with one of our patients, but I also put some quotes from some of our participants. A number of them spoke at the FDA Advisory Committee in support of this product being approved, and this is what they had to say. I can see better in low light. I can stay out later with my friends, playing outside. Now I feel part of the group. Gene therapy has made my world so much brighter. I'm able to see the stars, snow falling, fireworks, and most importantly, the moon. Within days, I could see vibrant colors again. I was no longer living in a black and white film. I can walk confidently in dim lit places. I went to a restaurant on my birthday and saw everything by candlelight. Next slide. And this slide is actually from the clinical trial. So you have a side-by-side video here. On the left is one of our participants at baseline. This is our primary outcome measure from the phase 3 trial. So this is the same light level on the left and right side, 1 year after on the right side. So the video is a little choppy on my side, but you can see a clear difference. She's running into obstacles before treatment. She's going really slowly, getting lost. And then on the right side, she's already done. She already finished with no collisions, no getting lost. And just very simply, I think it's like 20 seconds or something. So I think this video, as well as the quotes from the participants, really illustrate the impact that this product in particular and gene therapy just overall can have on the life of children and research participants. There's such great potential in ophthalmology specifically here, but in so many other areas of medicine.
Wonderful. Thank you, Sarah, for sharing this heartfelt story. You know, it's always nice to be reminded of the work we do. Sometimes we get stuck in the day-to-day, but it's nice to step back and see that what we do really makes a difference. And now we will hear from Dr. Cruz. He will provide a high-level introduction to cell and gene therapy and the promises it holds for the future. Welcome, Dr. Cruz.
Thanks again, Angie. So before we go through this overview about these exciting new therapies, let's talk just very briefly about our existing ones and where they fall short. So many chronic and genetic diseases lack a true cure right now. Treatments often focus on managing symptoms and slowing disease progression. And medications, it can be effective, but they have a lot of side effects and require long-term use. And these traditional approaches, you know, including surgery and these pharmaceuticals and sometimes radiation therapy, have revolutionized healthcare for sure, but they have, you know, limitations in terms of, you know, like the cells and gene disorders that we are currently facing. So in the field, we have a saying, evolution by natural selection outperforms intelligent design. Nature, after all, has had millions of years to find the answers, and we've only just started. And so as the slogans that you could see from cell and gene therapies facilities say, the cure is inside all of us, inside our genes and inside our cells. Next slide. So this is where cell and gene therapies come in. These revolutionary approaches offer a whole new way of thinking about treatment. We've witnessed remarkable resurgence in the past few years, and this period marks the culmination of decades of research, with more approved therapies finally reaching patients. So we'll hear of cures of blindness, cures for cancer, and these therapies address a wider range of diseases and targeting, you know, different tissues. Cell and gene therapies address some of the limitations seen in traditional therapies by offering a more targeted and potentially curative approach. They can potentially target the root cause of a disease by modifying the cells or genes directly and fix the underlying genetic or malfunctioning cellular disorders. By addressing these root causes, they have the potential to significantly improve the patient's quality of life. And this, in turn, reduces the need for repeated therapies and a potential for a lifelong, you know, cure resulting from this one therapy. Next slide. So this is where, though closely intertwined, there are clear distinguishing characteristics for cell and gene therapies. So cell therapies introduce cells into the patient, right? So they work with cells directly, and we usually use healthy cells or modified cells from the patients. And these cells can span multiple different tissue types. On the other hand, next slide. Gene therapies modify the genes inside the cells, right? So this can be given directly into the patient, and then it modifies them while inside the patient's body or in vivo. Or we could do this, in the laboratory or ex vivo and modify those cells and then introduce these cells into the patients. As we'll learn in a little bit, they use a lot of different vehicles or vectors to deliver the genetic cargo. Next slide. So let's talk about each of these therapies in turn, starting with cell therapy. So cell therapy aims to introduce healthy cells into the body to replace damaged or missing ones, potentially restoring function. Cell therapy is a cutting-edge approach that harnesses the power of healthy cells to treat a variety of diseases. The core concept is simple, introducing functional cells into the body to replace damaged, diseased, or missing ones. These introduced cells then take over the tasks that the original cells were unable to perform, and then consequently improving health. Next slide. So cell therapy can be broadly viewed in the schematic shown here. Cells are first obtained from patients or from other donors. They are modified in the laboratory, and then they are then reinfused back into the patient. So let's go through each component of the cycle. Doing this will give everyone here an idea of the areas of research, as well as the different moving parts that are responsible for successful implementation of these cellular approaches. Next slide. So there are two sources of cells for cell therapy. So you could get it from the patient directly or autologous, or you could get it from other sources or allogeneic. So autologous cell therapies, as we've said, use the patient's own cells. This avoids immune responses and, like the cells being recognized as foreign, but sometimes the quality of the patient's cells themselves, remember they have disease after all, limit the applicability of this approach. The complexity of manufacturing in real time, right? Like you can't manufacture them in advance of the patient coming into the hospital also limits some of these therapies. Allogeneic cell therapies, on the other hand, because it comes from another person, can be readily made. So they could be readily deployed when the patient comes in. And because they are manufactured in advance, this allows for larger scale production. However, because they are recognized as foreign, they could cause graft versus host disease due to the immune mismatch. Next slide. So some of the modifications that happen in the laboratory, right, like are necessary for many cell therapies, right? So whenever a patient's or another donor's cells are extracted, they're manipulated in this controlled environment before being reintroduced into the body. Some of these modifications include activation and expansion, where cells are extracted from the body, relatively inactive, and then adding signaling molecules like cytokines in specific culture conditions to stimulate them to both expand in number and increase their potency. We could also modify the genes of these cells. Various techniques can be used to introduce new functions into these cells to enhance their current capabilities or allow them to produce therapeutic molecules, as well as give them newer functions like migrating to sites where they would normally not migrate. And finally, differentiation, basically using physical and chemical cues in the environment so that we could create new kinds of cells from pre-existing cells. And this is often the route of stem cell therapies. For stem cell therapies, differentiation is essential to create the desired therapeutic and functional cell type, mimicking what's naturally occurring in the body. Next slide. So a few examples that you might have heard, include CAR T or chimeric antigen receptor therapy, where these cells have been redirected to recognize cancer. TILs or tumor-infiltrating lymphocytes, which have been obtained from the patient, grown a lot in the laboratory, and then reinfused more powerful than they have before. And also MSCs, or mesenchymal stromal cells, which are immunomodulatory cells derived from multiple different tissue sites and easy to work with in the lab and can be safely administered to patients. They've been used from various things from GVHD prevention to COVID cures. Next slide. So let's shift gears a little bit and talk about gene therapy. So gene therapy seeks to correct diseases by directly modifying a patient's genes, right? So these can be normal genes, or they could be corrective or trans genes. Next slide. As we have done for cell therapies, a schema, I think, is the best way to understand the landscape of gene therapies. In this, we see the different steps for gene therapies to exert their function. They're delivered to the cell, they introduce their modification, and they make new products. Next slide. Let's first talk about delivery. So delivery basically is the vehicle, how the gene gets into the cell. And there are two main ways. One is viral, right? Which basically piggybacks off nature's design, because viruses have evolved efficient ways to enter cells and deliver their genetic material. And scientists have used this function to basically, allow them access to the cell's inner workings. And then non-viral vectors often offer a safer option, but they're a little more cumbersome. So non-viral vectors, including nanoparticles, all these chemicals that encapsulate these genetic material, allow them to be introduced into the cell, although at a limited efficacy than viral vectors. Next slide. So gene therapy works by introducing several modifications, including gene replacement, replacing a faulty or missing gene with a healthy copy. Gene editing, so a more precise technique made possible by technologies like CRISPR-Cas9, and more recently base editing and prime editing, which can correct mutations, disrupt disease-causing genes, and insert new gene sequences. Also gene silencing, where you turn off or reduce the expression of specific genes, and promoter modifications, which alter the regulation of these genes' expressions. Next slide. And then finally, some of the current uses of gene therapy are shown here. We've seen gene therapies for sickle cell disease, where patients' quality of life has improved. Duchenne's muscular dystrophy, also the thalassemias and hemophilia, and inherited retinal disorders like Leber's congenital amaurosis, which has successfully restored vision in patients suffering from the genetic disorder. So back to you, Angie.
Thank you, Dr. Cruz, for that fantastic overview of cell and gene therapy. I know this probably only skims the surface of what we could learn, but I appreciate you giving us this dumbed down version for us non-science people. So now I would like to turn it over to Dan, who will share with us the current state of gene therapy, as well as the regulations in place to safeguard not only human subjects, but also those conducting the trials. Welcome, Dan.
Thanks, Angie. So the most common approaches we're seeing include gene-modified cellular therapies, genetic vaccines, gene transfer, oncolytics, which is predominantly about using viruses to kill cancer cells, gene editing, and use of gene-modified bacteria or bacteriophages, which are viruses that specifically infect bacteria. Next slide. This field is very quickly growing. This is a figure from a paper we published in Applied Biosafety, which was selected for the cover. It shows the number of investigational new drugs issued by the FDA specifically for gene therapy. Next. Not that long ago, the idea of having an FDA-approved gene therapy felt as impossible science fiction. The field has overcome multiple challenges, and we now see several approvals per year. Initially, oncology led the way. There was a surge of research and approval of infectious disease vaccines that coincided with the COVID pandemic. And we're currently seeing a surge in rare disease research, which will likely outpace approvals for all other disease indications. Next slide. This surge in rare disease research will predominantly affect pediatric subjects. The FDA has launched what was originally referred to as Operation Warp Speed for Rare Diseases and has since been renamed as the START program, or Support for Clinical Trials Advancing Rare Disease Therapeutics. Next. This program intends to make the FDA much more available to drug companies developing rare disease drugs, well in advance of conventional pre-IND meetings. They want to help with guidance on manufacturing, preclinical testing, and design of clinical trials. The FDA also hopes we have enough critical mass in the rare disease space to create a bespoke model. Their presentations often refer to the soda dispensers where a single technology can be utilized to deliver customized products to customers. They hope they can take advantage of using a small number of highly studied viruses with good safety profiles and create a pipeline where you simply select the gene of interest that would be therapeutic to a specific subject to produce the virus. The FDA hopes this will result in a massive surge in research and approvals. Now, I'd like to refer to a quote from Peter Marks, the director of the FDA Center for Biologics Evaluation and Research, or CBER, which reviews and approves gene therapies. I think it would be a shame if all we manage to do every year in the next few years is approve another 2 or 3 gene therapies. That's a failure, he said. Success would be that we start to watch what should be, if not exponential, at least some logarithmic progression here towards more and more gene therapies being approved. Next slide, please. So if we look at the FDA-approved gene therapy products for rare diseases, you'll see diversity in the types of technologies being utilized, as well as a diverse array of viruses used as gene delivery vehicles. So adeno-associated virus, or AAVs, are quite useful for delivering genes into non-dividing cells and tissues. Lentiviruses are commonly utilized to modify dividing cells such as bone marrow stem cells. Herpes simplex, or the cold sore virus, was actually the first approved viral vector as an oncolytic for melanoma, and that virus has since been highly attenuated and has been approved for topical application in epidermolysis bullosa. We also have our very first approved gene editing drug for sickle cell disease and beta thalassemia, and this involves electroporation of CRISPR-Cas gene editing technology into stem cells obtained from blood via leukapheresis. Next slide. So you may ask, why these investigational products involve viruses? So naked DNA by itself is not easily taken up by cells, especially human cells. So scientists look to nature to find better gene delivery vehicles. If we look at the viral life cycle, infection involves the delivery of the viral genome to host cells in order to take advantage of the host's biosynthetic machinery and allow viral replication. Next slide. However, if we remove the genes involved in viral replication and disease, the virus is only able to bind to host cells and deliver genetic material. Next. So we're left with what is essentially a genetic syringe, which we refer to as a viral vector. Viral vectors allow us to easily introduce genetic material into target cells during infection. While the viral vectors are typically unable to replicate, they are still infectious and must be handled accordingly, which I'll touch on later in this presentation when we deal with regulatory and logistical issues. Next slide. So this figure shows the various animal virus families. Viruses encompass a great deal of diversity, which gives potential for a large number of potential uses and risks. There are also diverse possibilities for toxicities. It's important to have a solid background in virology for review of virus-based studies. Next slide. So now we'll progress to regulatory considerations for cell and gene therapies. Next. Cell and gene therapies must undergo the same level of oversight as other drugs. Approvals are issued by the FDA Center for Biologics Evaluation and Research. While Early phase trials focus on establishing safety profiles and involve smaller number of subjects. Later phase trials focus increasingly on efficacy and involve progressively larger numbers of subjects. It can be quite challenging to find the appropriate number of subjects that qualify for studies, especially for later phase studies. Multi-site studies are common, especially for later phase studies. Next. So, of course, requirements for human subject protection still apply, which is provided by an Institutional Review Board or IRB. Research involving engineered genetic materials such as genetic vaccines, gene-modified cellular therapies, and gene therapies can require an additional level of review in the form of an Institutional Biosafety Committee or IBC. The IBC ensures compliance with NIH guidelines, which focus on assessing the risks associated with engineered genetic materials. The IBC focuses on protection of the research staff as well as the community and the environment around the research site. And this role is important considering that many cell and gene therapies are either comprised of infectious materials or utilize infectious materials during the manufacturing process. Next slide. The IRB and IBC should work collaboratively, especially when reviewing the informed consent document. The IBCs assess the risks associated with the genetically engineered investigational product and ensure the risks are adequately relayed to the research subject. The IBC can also assess risks pertaining to biodistribution and shedding from the research subject and recommend verbiage intended to protect casual contacts, intimate contacts, individuals who may be at increased risk of exposure, such as immune-compromised individuals or pregnant individuals, as well as children. Next slide, please. IBC review is required when two conditions are met. First, the research involves use of engineered genetic materials, such as in trials involving gene-based vaccines, gene-modified cellular therapies, or gene therapies. Secondly, IBC review is required when there are ties to support from the NIH. Support can be in the form of collaboration in developing the investigational product, or funding for the development of the study, or support of the research site. Even if there are no ties to NIH support, IBC review is recommended as a best practice and the NIH recommends voluntary compliance. Next slide, please. Each site must be registered with the NIH Office of Science Policy for them to be able to undergo IBC review. IBCs are comprised of at least 5 members who collectively possess the expertise to assess the risks associated with the engineered genetic materials under study. IBC membership must include at least 2 local unaffiliated community members to represent the interests of the community and the environment. And for each site, there is also a requirement for facility inspection to ensure proper containment and safety practices are involved for the proposed research. Next slide, please. Sites can register in advance of receiving a study to avoid administrative delays. Review of a site registration takes the NIH 6 to 8 weeks, which can pose a considerable delay to study startup. However, sites can register in advance of having a study requiring IBC review. In fact, this is what we recommend. For example, Advarra's Gene Therapy Ready Network is the world's largest network of sites registered with the NIH, which are ready to undergo IBC review once a study is submitted. Being IBC ready can also pose as an advantage during site selection. And sites can get registered with the NIH either independently or utilize an IBC service to perform the task for them. Next slide, please. I'd also like to mention the evolving regulatory landscape. The NIH imposed a single IRB mandate, which has also been incorporated into the common rule and will be required by the FDA. Next. So how do we accommodate the requirement for single IRB review while the NIH calls for local oversight of research involving engineered genetic materials at the site by the IBC? Next. 2 approaches predominate. The first is to utilize a single IRB with the bandwidth to service additional sites. However, this approach minimizes or eliminates crosstalk between the single IRB and the site's IBCs. The second approach involves utilizing a central IRB with an associated IBC service. This approach provides all the efficiencies and benefits seen in centralized review, including standardized forms, policies, procedures across sites and institutions. This also allows for each site's for crosstalk between each site's IBC and the IRB service, and it ensures that each site's IBC has the requisite expertise to review clinical trials, as many academic institutions with local IBCs focus on preclinical or basic science research involving laboratories and research animals. This approach, as I mentioned, also ensures crosstalk between the single IRB and the sites utilizing the associated IBC service. Next slide. So now we'll move on to logistical considerations. Next slide, please.
Give me just a minute, Dan, because I had operational considerations as going next.
Oh, I'm sorry.
That's okay. Sorry about that. We'll now turn it over to Sarah to share the site's perspective on the changing cell and gene therapy landscape and the impact on clinical trial operations. Sarah, the floor is yours. Oh, you're on mute.
Thank you. Sorry about that. And thank you to my fellow panelists for setting up a great stage for talking about how this affects us as sites. If you can next slide, please. So one thing we hear in the field of gene therapy quite frequently is the tidal wave is coming. And that is absolutely true, but I think in a lot of respects, the tidal wave is already here. This image I really like because it very clearly shows the breadth and the depth of the field. It's really busy, so I'll walk you through what it actually shows, but the bullseye on the outside shows all of the gene therapy clinical trials in the phase 1-2 stage. And then as you move in, you see pivotal, and then further in is those who are filed. And then on the very inside are the marketed products. So the figure also shows the breadth of gene therapy because it shows all of the different target areas. So on this figure, you have dermatology, hematology, metabolic, neurology, ophthalmology, otology, and then a very large other section up in the top left corner. And this image is actually a couple of years old. So if it were updated for today, there would be even more in every ring of the bullseye. The phase 1-2 trials that are on here, many of them have moved into the pivotal stage, and there are a lot more that have been added into the phase 1-2 session. Next slide. So this growth that we see in the bullseye there is actually reflected in the data of trials that have been launched in the past 4 years. So this is data from the American Society of Gene and Cell Therapy starting in 2021 and then the first quarter of this year. So as you can see, we have over 50 gene therapy trials launched so far this year. So we are definitely on pace to match or surpass 2023, the last year. And then I also like this quote from Scott Gottlieb, the former FDA commissioner, talking about the number, predicting that we'll have 200 INDs submitted by the year 2020. Next slide, please. And then this cumulative product launches. This is a predictive graph, and the quote from Scott Gottlieb continues here, predicting that FDA will be approving 10 to 20 cell and gene therapy products per year by 2025. And the predictive graph, of course, covers that as well. And then you can look at what they're looking at in the field by 2030. And I think we are right on target in 2024. I think we've had 7 products approved so far this year, and many more in the pipeline for approval. Next slide. And then, so focusing just at the site. So this is CHOP, and this is our in vivo gene therapy group at CHOP. We have, of course, seen growth here at the site comparable to what you're seeing nationally. These are the numbers just for in vivo, so this doesn't include any cell therapy. But you can see in 2021, we had 8 clinical trials for in vivo gene therapy, and this year we have 20, as well as 14 observational or natural history studies. And then breaking it down by divisions and indications, you can also see the growth in the breadth of the field. In 2021, we had 3 divisions, 4 PIs, and 7 indications. And then that's grown to 7 divisions, 14 PIs, and 15 indications this year. Next slide. So with all of that in mind, there are a lot of things that we have to consider on the site side of things. The first is rare and ultra-rare disorders, which we've talked about a little bit. With gene therapy comes the hope for treatments for rare and ultra-rare disorders. They're often overlooked by conventional trials, and it's really exciting to have hope for these patients that someone might be developing a treatment, even for an ultra-rare disorder. However, with rare and ultra-rare disorders comes a lot of challenges. You have a very small patient population, which makes trial design difficult with small n's. It also makes recruitment difficult. And of course, it's all very expensive and funding is hard to come by. And then if we're looking at in vivo gene therapy, it's a one-time delivery. This is great for patient burden because they're not having to take a medication all the time. They don't need to come to the hospital for regular infusions. But with that comes some drawbacks that you can't stop the drug. You can't remove it. So once it's been delivered, it's there. And then I think we're going to be talking about antibodies a little bit later, but with in vivo gene therapy with a viral vector, oftentimes you're going to develop antibodies, which means you can't get gene therapy again. So if one product doesn't work, you don't have the option of trying a different one that might come out later. Next slide, please. And then, of course, we need to talk about risks. There's a lot of risks of gene therapy, a lot of known risks, including very serious risks, but there are also a lot of unknown risks, particularly in the long term. It's a very new field. We just don't have long-term data yet. And as a rapidly evolving field, the data is always changing. So we are always looking at what's going on in the field and what do our patients need to know about what's going on in the field in order to make a decision about whether they're going to receive this drug or if they already have what they need to be looking out for. And then equity and access, of course, is something we need to be thinking about all the time, but it becomes even more important in the gene therapy space. In gene therapy research in clinical trials, the burden is extremely high on trial participants and their families. Trials are often run at large academic centers, which may require significant travel, even international travel. And then there's a large time commitment, which is also a barrier for families because they have families, other children that they need to take care of, or parents, they have jobs. So committing the time to a clinical trial with the intense follow-up required for gene therapy is difficult. And then there's a knowledge scientific barrier for families to understand gene therapy. And then with approved products comes an entirely separate issue of cost and insurance approval that we will not go into today, but it is something that we need to be monitoring in the field. And then all of this leads to the importance of informed consent. So of course, we all know the importance of informed consent in clinical trials, and I think it's especially important in gene therapy clinical trials because of all the considerations I just listed. And it's equally important for commercial products, approved products. So all of this requires provider education. Whoever is consenting patients needs to understand the science and the risks in order to properly consent. I would also argue that coordinators and other managing staff, people managing the day-to-day of the trials, need this education. Anyone who has worked in a clinical trial knows that participants often ask coordinators questions that they won't ask the PI. They've developed relationships with the coordinators and the day-to-day staff, and they feel comfortable talking to them. So I think this underlines the importance of educating the day-to-day staff. they need to also understand the science in order to ask questions and advise the families who are participating in the trials. And then I think I'm throwing it to Dan.
Yep, thanks, Sarah. Wanted to just share that's a lot of different considerations, especially when you compare to how much you have to think about with drug and device trials. And now I will turn it over to Dr. Cruz. I will find his slides, I'm so sorry, who will share about the scientific considerations of conducting cell and gene therapy. Knowing that we have about 40 minutes left, I will attempt to make our way through the rest of the presentation. Welcome, Dr. Cruz.
Great. Thanks again, Angie. So I'll be brief. As you have hopefully seen so far, there's a lot of promise from cell gene therapies, but it's worth noting that there's a lot of work to be done, including in the scientific area. So a common focus of research for both cell and gene therapies involve research into the specificity and the safety of these products. So if we talk about specificity, we're talking about ensuring that the therapy reaches the right cell type, the right environment, and all the relevant cells within the target tissue, right? So while avoiding off-target effects on healthy cells. For cells, this means we take a look at the off-target and unexpected results of the cells themselves. And for genes, this means we look at enhancing the tropism, or where the gene targets of the genes themselves and their vectors. For both approaches, we also focus on the potential of, these gene modifications to cause malignancy and other consequences of putting in new blueprints and new instructions to these biological therapies. So, because these cells and genes are essentially foreign, they're also prime targets for interactions with the immune response. And so there's also a big research focus on both minimizing the unintended immune response to these cells, as well as maximizing the ability of these cells and genes to persist in spite of the immune component. Next slide. Next slide, Angie. Yeah. So for cell therapy specifically, some of the ongoing research is focused on ensuring and understanding the migration where, delivering these cells effectively relies on them going to the site on their own. Persistence, ensuring that these cells stay long enough to perform their desired functions. As we talked about earlier, focusing on immune rejection, particularly for allogeneic cell therapies, where this is more pronounced. And unanticipated mechanisms of action. Remember, these are biological products. While we harness the therapeutic benefits of one of their many functions, they also perform a lot of different ones, and we have to be on the lookout for all these other consequences. Next slide. And then finally, for gene therapies themselves, ongoing research is focused on ensuring and understanding the risks, right? So creating safe and efficient vectors with minimal risks of insertional mutagenesis or unintended, insertions of these genes that will cause problems like cancer down the road. Developing vectors that target the correct cells, so altering the natural targets of some of these viral vectors. We also do research on method optimization so that we choose the most efficient delivery method for each disease and cell type. And ensuring that there is appropriate levels of gene expression, right, like that we deliver for these cells when we make these blueprints. Okay, back to you, Angie.
Thank you, Dr. Cruz. I now will turn it over to Dan to talk about logistics and the considerations of the difference between traditional trials and gene therapy. Dan, the floor is yours.
Great, thank you. Next slide, please. So there we go. So research involving cell therapies and gene therapies inherently involves work with biologics, and research involving biologics pose some unique challenges. For example, biologics are inherently less stable than small molecules and require special consideration for shipping and storage. While small molecules pose the risks for chemical toxicities, biological toxicities can be more complex and potentially prolonged. There is an inherent need for risk assessment to determine the risks associated with use of the investigational product for the research staff, as well as the potential toxicities to the research subjects. Next slide, please. So things to consider while preparing a site to conduct a gene therapy research is the appropriateness of their facilities as well as personnel training and procedures for the life of the investigational product. So this includes everything from shipping, storage, dispensing, transport to the clinic, administration, and waste disposal. This can include matters such as procedures for return or disposal of the IP. Is there potential for the research subject to shed potentially infectious materials? Does the facility have adequate design to conduct the research as well as the appropriate equipment and personal protective equipment for personnel to avoid occupational exposures while conducting the trial? Next slide. So consider during the rollout of the COVID vaccines, many sites lacked ultra-low -80-degree freezers and feared being unable to administer mRNA-based COVID vaccines. Temperature requirements led to shortages of dry ice. Next slide. And supply chain obstacles hampered the rollout of the COVID vaccines. Next slide. One common issue to consider when looking at pharmacies is if they have the appropriate equipment for dispensing the investigational product. For example, many sites confuse biosafety cabinets and clean benches. But as you'll see soon enough, all hoods are not created equal, and this can impact the pharmacy's ability to dispense cell and gene therapies. So what you're seeing here is the airflow diagram of a clean bench. Clean benches blow HEPA-filtered air across the work surface to provide product protection. However, they're not intended for use with any type of hazardous material, including infectious agents. In fact, clean benches increase the risk of occupational exposure to the pharmacist as they blow towards the worker and out into the pharmacy. Next slide, please. Biosafety cabinets are recommended for cell and gene therapies as they provide protection to both the product and the personnel. Biosafety cabinets HEPA filter sterilize the air blown into the work area. Once it's potentially contaminated with exposure to the investigational product, the air is then sucked back into the hood and blown through a second HEPA filter before being exhausted out. This protects the worker, the facility, and the environment. Next slide, please. Also consider while shared infusion areas are designed for comfort, they are not designed for working with infectious materials. A spill could expose other patients or research subjects who did not consent to the exposure and may be at increased risk of exposure to infectious agents as they may be undergoing treatment with immune suppressive therapy regimens. Next slide. As decentralized trials become increasingly common, IBC review is important to ensure all studies utilize appropriate facilities and procedures. The IBC can work with sites to design risk mitigation strategies as challenges arise. Next slide, please. So some examples of common issues that arise in healthcare facilities. So for example, how to restrict access if using bays separated by curtains. This may be appropriate if materials can be transmitted by splatter, but not if aerosol transmission is a concern. Personnel must be familiar with how to handle spills. Biologics must be disinfected rather than cleaned up with a chemo protocol, and using an inappropriate disinfection strategy can just spread contamination around the research site. An appropriate plan must be in place for handling occupational exposures. The site's bloodborne pathogen exposure plan often serves as a good model or starting point to developing an appropriate exposure plan for the investigational product. When handling the investigational product, access should be limited to the minimum number of personnel required for the study. So, how do we go about restricting access in these situations? A related concern, especially for pediatric trials, is protection of the parents while a pediatric subject is receiving the investigational product. Oftentimes, it may be helpful to have the parent in the room to help the pediatric subject relax while receiving the investigational product. If the parent is in the room, they should receive hazard communication to be made aware of the risks, as well as receiving personal protective equipment comparable to that worn by the research staff to protect them from exposure from a possible spill during administration or from the inoculation site. So consider that a child receives the investigational product, and then maybe they want to cry or hug their parent, or there's leaking from the inoculation site. And you could see how this could easily get onto the parent or the parent's clothes. It's also worth considering whether the children need to be kept in a separate room for observation rather than maybe going to a waiting room or play area where they could potentially contaminate the waiting room, toys that could be utilized by other children or come into contact with other children. Next slide.
I think that wraps it up for you.
Yep.
There certainly are a lot of logistics to consider before embarking on the cell and gene therapy. Now we'll hear from Sarah. She's going to talk about the infrastructure. There's been some questions about that posted in the chat to do the cell and gene therapy at the site. Sarah?
Yeah, thanks, Angie. So I think on the site side, of course, we are constantly assessing infrastructure, both physical infrastructure and personnel, because of all those things that Dan just outlined. Also comparing gene and cell therapy, there's some overlapping needs, and then there are some where the needs diverge. So there are universal clinical needs for both for conducting a trial in either one. So we need spaces for consent and any applicable evaluations. That could be radiology, physical therapy, ophthalmology, audiology, or phlebotomy, or any other number of locations based on the trial. But of course, we have to balance this with the demands of an active hospital or medical center. There's clinical demands on all of these spaces, and we have to figure out how to balance them. And then we differ a little bit in between gene and cell therapy when we're talking about the drug preparation and administration. So cell therapy, you need apheresis and collection and handling. You need a place for product storage and a location for infusion. And at CHOP, all of these cell therapy products are run through the transplant division. For gene therapy, you need pharmacy storage and you need sterile drug preparation that Dan was talking about. And then you need an administration location, whether that's the OR or that's an infusion suite, or whether that's radiology for an LP or an ICM injection, depending on the trial design. You need staffing for long-term data collection. And then the difference with gene therapy, specifically at CHOP, is that these are all run across multiple clinical divisions. Next slide, please. So because of the diverse needs of gene therapy at CHOP, we founded in 2021 the Clinical in Vivo Gene Therapy Group. So this group provides regulatory support, scientific and operational support to PIs regardless of their clinical division. We have a staffing core of CRCs and research assistants, as well as regulatory and administrative personnel who specialize in gene therapy. They are available to any PI doing gene therapy at the institution. We support clinical trials, observational and registry studies. We also help develop investigator-initiated studies and support pre-IND and IND writing. We've also created a gene team, which is an on-call multidisciplinary team of experts who can be called in to consult on any suspected gene therapy adverse reaction. And this is both research and clinical delivery of gene therapy. We also have an internal registry and biorepository to look at long-term outcomes of gene therapy. We have a clearinghouse to onboard licensed therapies. We have an educational symposium that we're putting on in September to educate the larger community. And then we've also developed and implemented gene therapy shedding precautions. So this is during administration as well as after administration. We have institutional requirements on PPE, cleaning, and contact with others. And we also have recommendations for households. Same idea, PPE cleaning, particularly contact with other affected siblings or family members. And then we also have quarterly investigator meetings that are open to any gene therapy PI at the institution. So all of these initiatives are helping us break down the barriers that we identified between divisions so that gene therapy investigators are connected to each other and there's consistency and safety throughout the institution.
Amazing,
Thanks, Angie.
Sarah. I definitely think that your site would be the gold standard. I think we could learn a lot from CHOP about the requirements, and your program is just very impressive. So next, I'd like to move into some questions. We have quite a few that are posted. I think the first question, maybe Sarah can field, and then maybe Dan can kind of jump in, but it's looking at that burden of the long-term follow-up, right? So gene therapy is one and done, but then how long do you follow them? 15, 20, 30 years? Like, what's the requirement? And if they become adults, you know, do they have to reconsent? Like, how do you manage that?
Yeah, absolutely. So the regulations differ depending on what the product is, but it's between 5 and 15 years FDA required follow-up for an investigational gene therapy product. It's certainly a burden. It's something that we're constantly thinking about at the site. How do we support 15 years of data collection with, you know, staff turnover and all of those things? And that's one of the things that we've tried to implement here at CIGT is to make sure that we have that consistency. To answer your question about children who turn 18, yes, they do have to reconsent, which is always an interesting step, making sure that they are educated. You know, hopefully they could provided assent previously, but so they're fully informed on the trial. But you never know, with an 18-year-old, they might decide to drop out.
And Dan, how do you handle that? I mean, in terms of collecting safety data, you know, how does the IBC look at that? And then, you know, how do you determine how long they have to be followed? Is that a decision made by you or the protocol?
So the FDA provides guidance on that, and in biosafety, so with several of the questions that I'm seeing in the chat, biosafety is risk assessment based. We have to look at the nature of the investigational product. What are some potential toxicities associated with that investigational product, and the appropriate time frame for follow-up. So if something, for example, is an integrating vector, if it has the ability to insert itself into the DNA of the host cells and potentially introduce a mutation, then there tends to be longer follow-up because of concerns over long-term consequences like the development of cancer over time. So, and that's where Sarah mentioned the 5 to 15-year follow-up. Now, recently in the news, there have been a number of secondary lymphomas associated with the use of chimeric antigen receptor T cells that are being investigated by the FDA. And they've actually added box labeling and also extended it, the long-term follow-up to the life of the research subject. Because of these concerns. And they do advise that sponsors propose a plan for the extended long-term follow-up of these research subjects, even after the study closes. As far as how to do it, that's ultimately up to the sponsors.
Wonderful. Well, unfortunately, we have time for one more question. There's been a lot of great questions posed, and I'm sorry that we didn't get to all of them. One of the questions, and I had never thought about this in pediatrics, the parents being exposed. One of the questions is, you know, how long do they have to wear PPE? Would it be just administration? Would it be follow-up appointments? What are the requirements for that?
So this goes back to the risk assessment associated with the investigational product. And we can look at that in terms of some of the data that will be in the investigator's brochure. So the IBC will review it. And that's one of the benefits of IBC review versus IRB. We look at the risks to the research staff as well as everyone that the research subject could come in contact with. So we'll look at biodistribution and shedding and provide recommendations regarding, for example, does the subject need to be under observation in isolation from other children, for example, for a certain amount of time? When handling specimens from this individual, do they need to have added level of protective equipment on top of what they would normally wear? as part of just their standard procedures. Unfortunately, I can't give a one-size-fits-all answer. In biosafety, everything is risk assessment based.
Wonderful. So we have to end today, but we hope today's webinar has highlighted the rapidly growing landscape of cell and gene therapy and provided you a deeper understanding of this complex science, the need for specific type of infrastructures, and lastly, the importance of understanding the regulatory requirements and safeguards. Thank you for joining us today and being a part of this important conversation. So a few housekeeping announcements before you sign off. We will have a webinar about artificial intelligence being used to transform pediatric clinical trials. We will look at how it's being used to make more efficient, accurate, safer, and cost-effective studies. The next webinar will take a deeper dive into cell and gene therapy, maybe answer some of the questions that were posed. We'll look at current treatments and corrective therapies. We'll identify the regulatory challenges such as toxicities, and then we'll look at ways to mitigate those challenges. If you would like to receive CE credits for today's webinar, please reach out to us at this email address, and you can be provided with a certificate of attendance. And if you are interested in today's webinar, you can find the recording and slides on our site network portal. I did see somebody post about having challenges getting into that portal. Please reach out to us and we can help you facilitate that, but they also will be sent out. The good thing about the portal is you can access webinars from our previous series. So there's a lot of good information in there. Lastly, please take a minute to fill out the feedback from today's webinar. We are continually looking at ways to improve our delivery of our educational programming. And finally, we want to recognize and thank Gilead Science for their support with our educational webinars. We are grateful for their commitment to pediatric research and education. Thank you, everyone, and goodbye.